game. So as you can see, I changed the title a bit compared what you find in your program. So just to highlight a few key points of my talk. What I'm going to talk about is 4sU-seq, sort of a new RNA-seq technique, which allows us to analyze RNA splicing and processing in more detail. So all of the stuff I'm going to talk about now is actually contained in this paper from Genome Research, so now you know why I shortened the title. There's a lot of other stuff in there which I'm not going to talk about today in the interest of time, but I hope you'll read the paper anyway. So the method we use 4sU-seq applies first a method called 4sU-tagging for labeling newly transcribed RNA, and the way it works in short is that you use 4thiouridine, which is can be applied to cell cultures or also in vivo. And the result is that in some way the newly transcribed RNA is labeled, and in the uracils which are incorporated during the de novo transcription, they have a thiol group attached to them which can be used to purify the newly transcribed RNA and to separate it from the pre-existing RNA which is left over from the beginning of labeling. And then you can quantify this RNA, both the newly transcribed RNA and the pre-existing RNA and also total RNA, either with microarrays, this is what we did previously, or using RNA-seq, and then it's 4sU-seq. So that's basically it. And this gives you simultaneous measurements of RNA decay and de novo transcription. So previously, we already did a few studies using this RNA tagging method. Previously you used microarrays, but as you all know is that if you use microarrays, you only get one value per gene, or if you use exon arrays, you get only some parts of the gene which are probed. Whereas if you do sequencing of RNA, you actually get the expressed parts of the gene, at least those which sequence well. And if you combine this with tagging of newly transcribed RNA, you can actually see that the newly transcribed RNA is not yet completely processed. So if you combine these two techniques, RNA tagging and RNA-seq, you can actually look at the processing of the RNAs. So usually if you do RNA-seq, so it's just an overview of the RNA-seq protocol, which most of you should know. You get your sequence reads, and usually you get them from the exonic parts and the exon-exon junctions. If you do 4sU-seq, you also get intronic reads of partially spliced or not spliced transcripts, and you get also exon-intron junction reads. And you have to include this in your mapping process, but that's not a problem really. So previous studies that combined 4sU tagging and RNA-seq, they always used relatively long durations of tagging, so 45 minutes, that was I think the first study who did this, and a later study used 2 hours. And as we will later see at this point, most of the newly transcribed RNAs are actually already spliced, so you don't really can look at splicing or processing. What we did is we used ultra-short RNA tagging. So we labeled for the first 5 minutes, the first 10 minutes, the first 15 minutes, 20 minutes, and then 60 minutes. And we also extracted total RNA and the pre-existing RNA at 60 minutes. So what we get is RNA samples with increasing age of RNA. So the 5-minute 4sU samples, the RNA in there is at most 5 minutes old. In the 10-minute 4sU sample, it's at most 10 minutes old, and so on and so on. In the total RNA sample, you have all the RNA in the cell, and if you look at the pre-existing or untagged sample, you get the old RNA, which is at least 60 minutes old. And what we saw is that if we sequence these samples, we can actually visualize splicing all the transcripts. So these are just the distribution of the different types of reads we get, so intronic reads, exon-intron junction reads, exon-exon junction reads, and exonic reads. And on the x-axis, you have the duration of labeling, or the age of the RNA. And as you can see, the contribution of intronic reads decreases slowly, whereas the contribution of exonic reads and exon-exon junction reads increases. So what you see is that here splicing is happening, the introns are spliced out, and most of the transcripts at this time contain only exonic parts. There are still quite a lot of reads from introns, but the reason for this is that the introns are so long that even if you just have a few reads every other 100 base pairs, you still get quite a lot of reads. So this last column here is actually not a sample, but these are the estimated frequencies for completely unspliced transcripts. And if you compare this to the 5-minute RNA samples, you see that actually there is quite a difference between the frequency of intronic reads between the sample and the completely unspliced RNAs. So at first, here we have in a 5-minute 4sU RNA, we have around 76%, whereas the expected frequency in completely unspliced is 89%. At first, this doesn't look too differently, but we use this to estimate the fraction of introns which are actually already spliced at this point. And we estimated that at least 65%, and this is a very conservative estimate, of introns are already spliced at this point. So we really need to look at these ultra-short RNA tagging durations to still be able to really look at splicing and processing of RNAs. So what we also did is, because before we could not really see these junction reads because the relative contribution was so little, what we did is we normalized the frequencies to the maximum frequency in any sample, so that we compare the change in intronic reads and exon-intron junctions, or on the other hand, the exonic reads and the exon-exon junctions. And what you see here is something which actually at first made our biologists very excited, because there seemed to be a delay between the decrease in introns and the decrease in exon-intron junctions. So it appeared that the introns were already decayed while the exon-intron junctions were not yet spliced. But when we looked at introns with similar lengths, we saw actually that there is no such delay in each of these groups. Each group has approximately the same size, and you see that there is no delay here or here. But what you also see is that actually the curve is much steeper here than here. And this is actually what we found, that for some reason longer introns disappear faster, and because they contribute more reads to the intronic reads, but not so much to the exon-intron junction reads, because there's no matter how long the intron, the number of exon-intron junction reads it can produce is the same. Whereas, of course, if you have a long intron, it can contribute very many intronic reads. So this is just the reason for this strange effect. So what we saw is that apparently the intron length and the decay rate were in some way correlated. So what we did is, to analyze this in more detail, is we calculated the 60-minute to 5-minute ratio. So this is the intron level in a 60-minute 4sU RNA, which is a bit older than the 5-minute 4sU RNA. And if this ratio is high, then the intron is still there in relatively high values, at relatively high values, whereas if this ratio is low, then it has already been mostly decayed. And what we saw is that, as expected, the longer the intron, the longer it did appear to disappear. But there was actually a peak in the medium intron length range with even faster decay rates for the very short introns. So we don't really have an explanation for this, but this is what we saw. And this is not due to the fact that longer introns are contained in longer genes, which we also saw that introns from longer genes also did appear to be decayed faster. But again, if we separated the introns into groups of approximately the same length, we saw that within each group the same pattern was observed. So the correlation between gene length and decay rates did not explain the correlation between intron length and decay rate. So this is the figure I showed you before with this delay. If you normalize to the intron length, the delay basically disappears. So it does not appear that there is any, that the introns are actually decayed during splicing. So another factor which we thought might influence the decay rates is the splice site strength. So we did the same analysis. We analyzed splice site strength, predicted splice site strength for these introns, correlated this to the intron decay rates. But here we did not really see such a strong effect. It was mostly for very weak splice sites that they were decayed more slowly. And again, if we group the introns according to their length, we saw that this effect was mostly observed for very short introns and very weak splice sites. So here the effect is not this strong, really. So this was a global analysis looking at all introns. What we did then is to look at individual genes, individual introns, but we only looked at the most highly expressed genes, because we need a lot of reads to actually do a reliable analysis. And since even at 5-minute 4SU RNA, a lot of the introns are already spliced at very low levels, we only expected, well, acceptable numbers of reads for these introns if the genes were actually very highly expressed. So this is why we focused on the most highly expressed genes. We had 525 genes in total, and around 7,000 introns for this analysis. And for about 1,000 of these, 15%, we saw that they were actually already absent in the 5-minute 4SU RNA, in the very newly transcribed RNA. We never saw them. And of course, there's a lot of artifacts which can produce this. There's problems with, might be problems with sequencing these parts. But we found, at least in some of these cases, that the exons on both sides of these introns were really clearly expressed. And so it appeared there was actually some very fast splicing happening. And one example which shows this very nicely is this gene, which is actually a SnoRNA precursor. And for this gene, there is a known retained intron, which is this green part. And this lilac part is known to be annotated to be spliced out. And we also saw that this part is absent in 5-minute 4SU RNA, so basically it's not even there in the youngest RNA sample, but the rest of the known retained intron is actually still there, and we see it until 60-minute 4SU RNA. So it seems that this transcript, containing a part of this intron as a retained intron, is really actively produced during splicing. And what we also saw is that for these absent introns, the frequency of exon-exon junctions was not increased. So if they were very spliced very rapidly, we would expect that the exon-exon junctions are then there and are increased compared to other introns, but we did not see this. And the hypothesis is that actually here there's an intron decay concurrent with splicing, and there is also a paper which shows that this can happen, that exon tethering happens, so that the intron is already spliced while the intron junctions are still connected to the splicing machinery. So the next thing we did is basically a clustering of the introns to see if there are different patterns we can observe. And what we used for this is the, cannot really read, it's the intron to gene ratio. It's basically the intron expression level compared to the overall gene expression level, and kind of gives you the fraction of transcripts which still contain this intron at this point. And well, you see, I hope you see at the first glance, there are kind of different patterns. So these two look very similar. The only thing which is different is the level at you have here, and there is a number of other clusters which look similar. So what we did is we clustered these clusters again to define 4 types of classes which show a similar pattern. And these are just the box plots for each of these classes. And the first class is the one with the highest number of introns, which we then denoted. This is the normal decay class. So this is how, for most introns, decay happens. And you see it goes down approximately linearly in the first 20 minutes. So this is what happens in most of the cases. Then there's a second class, which is second largest class, where we see that it goes a bit slower here, so there's sort of a bulge where the introns are not decayed that fast. Then there's a third class where we actually see that between 5 and 10 minutes there's a large decrease in the exon to gene ratios. So this appears that here's a very fast decay. And then there is the last class, which we thought was very interesting because here the intron to gene ratios hardly change in the first 60 minutes even, but they do decrease very rapidly at the end in the very young, old RNA samples. So what some of the reviewers criticize is that, of course, we only looked at the most highly expressed genes. We do not know if this pattern is also observed in less highly expressed genes. So what we did is we took all the introns from the other genes and tried to assign it to the most similar class. And this is how the plots look if you use all of these introns, and you can see that the pattern is pretty much the same as here. What changes a bit is the relative frequency of each class. So if you look at all introns, we actually see that the normal decay class is a little bit less frequent, and the other class is a bit more frequent, but which is the class which is most frequent, the class which is less frequent, this doesn't change. So now I just want to give you an example how this looks for individual introns. So here's an example for an intron of class 1. And you see that the number of reads, so the color and the bars indicate the reads and the number of reads. And you see that these numbers sort of decrease from 5 minutes to total and untagged RNA. So this is an example of class 4, where we actually see that, well, these levels do not really decrease. And even at 60 minutes, we still have quite a lot of reads. But in total and untagged RNA, they're basically gone. And maybe this reminds you of one of the pictures I showed you before, the example of a known retained intron, where we also have this case that the reads are there until 60 minutes 4sU RNA, and then they are basically gone. So we hypothesize that this class 4 might actually consist of new examples of intron retention events. But if these are really intron retention events, the interesting stuff about this is that actually these seem to be gone in total and untagged RNA. So they are no longer present in old RNA samples. So we wondered if this kind of temporary intron retention is something we actually also see for known intron retention examples. So we looked at exons annotated in ensemble transcripts. We classified them into 3 types of exons. So core exons, which are basically there in almost all of the transcripts. Then intron retention examples and alternative 3' and 5' ends. And what you see here, if you look at the core exon examples, you see that the relative ratios, so the number of fraction of genes which contain these exons, don't really change across the RNA samples. So they're in the 5-minute 4sU RNA, and they're still there in untagged RNA. Whereas for the intron retention cases, we actually see a very similar pattern. The levels are stable, at least they don't decrease that rapidly in the first 60 minutes, but in total and untagged RNA, they're basically gone. And this is also seen for alternative 3' and 5' ends, but not as strongly. So it appears that actually retained introns are produced pretty regularly in around 2 to 8% of cases, but they're not very stable. So they only have short-term stability, and at some point they are decayed. But we don't know yet how, so that might be that we have either a nonsense-mediated decay of the whole transcript, or there might be some sort of secondary splicing event which just removes this intron. So what we also looked at this is other properties of splicing classes, because we already saw that intron length and gene length and splice site scores were somehow correlated to the decay rate. So we looked at these values for each class. And you see here, so this is always the ratio of the median class value divided by the median of the other classes, and we take the logarithm of this. So a 0 means there is no really difference to the other classes. A positive value means that the value is increased, a negative value that is decreased. So what you see is, this is again the normal class. the class with slow decay, fast decay, and the very slow decay, the potential intron retention events. And what you see is that this class has the very longest introns, whereas these are very relatively short introns. The same is true about gene length. So these are the longer genes, these are the shorter genes. And then you see that both the slow and the very slow decay classes, they have weaker splice sites. So here and here, this is a bit decreased. And we can differentiate between the two classes because these are short introns and these are very short introns. So it seems that the classes are characterized by distinct combinations of intron length, gene length, and splice site strength. And just one short example I also want to give, which is discussed in much more detail in the paper. We also looked at SnoRNA processing. And because SnoRNAs are encoded within introns of other genes, and they're just processed out of these introns during splicing. And what we saw is that SnoRNA precursor introns are actually enriched in the class 2 and class 4 examples, the ones where we saw relatively slow decay. And they're de-enriched in class 1, in the class with the normal decay. And so we just looked at the same curves for SnoRNA precursor introns and remaining introns. And what you see is that the remaining introns are processed or decayed faster than the SnoRNA precursor introns. And if you put in a line, you see, well, it's basically just linear in both cases. So this is just slower. We don't really see the same bulge we saw in classes 2 and 4. So it appears that this is not an intron retention followed by decay, but the hypothesis we have is that there is actually a competition between SnoRNA processing and the splicing machinery, which kind of slows down the processing at this point. So then I'm already at the end of my talk. Just a few conclusions. What I showed you is that ultra-short RNA tagging is compatible with RNA-seq. The newly transcribed RNA already still contains large fractions of unspliced RNA, but so the combination with RNA-seq allows the analysis of RNA processing at the nucleotide level. But we also saw that in 5-minute 4sU RNA, at least 65% of introns are already spliced, so we really need to look at these very short RNA tagging durations to analyze RNA processing. And yeah, that's what I just said. And what I also showed is that it allows the identification of distinct classes of intron splicing kinetics, and even the identification of potential novel intron retention events, which have short-term stability but are eventually decayed. So here are just a few references. So this is an older paper on RNA tagging. This is the genome research paper, and a new method we developed for mapping, and a new paper we just got accepted, which uses these mapping algorithms for metatranscriptomics. And just a few acknowledgements. So I really need to acknowledge Lars, because he's the one who basically pushed this method forward, and collaborators from Ralph's group and from my group, and of course I need to acknowledge funding from the DFG. So thank you for your attention. I'll be happy to answer any questions now.
Short question. Do you explicitly address or include alternative splicing in your analysis? Because if you have multiple isoforms present in the same cell type now, you wouldn't quite know if you should count this as exon or intron. I want to bin it in specific intervals, or explicitly try to deconvolve it.
Yeah, well, we always looked at introns which were never annotated to be in a transcript. So if there is one transcript containing this part of the intron, we count it as an exon and only looked at it in this analysis where we wanted to see if it's also for known alternative splicing events. Otherwise, an intron was something which is never contained in a transcript. Of course, we can't really differentiate between other unknown alternative splicing events. You mean minor with alternative splicing isoforms? No, we did not really look at the ratios of frequency of each. of alternative splicing in a sense. But as I just said, we basically did not look at the unknown transcripts, we just looked at the introns not known to be in any transcript so far.
Do you think you could observe recursive splicing? Did you observe this case with recursive splicing?
The sequencing depth was not high enough to really distinguish this. So we wanted to, but we figured out that this was a pilot study, so we did not really have this high sequencing depth. So we didn't have the resolution to really distinguish between nonsense-mediated decay and secondary splicing. But this is, of course, something we really want to look into.
Actually, do you assume that splicing is always co-transcriptional here, or?
No. No, we could have. So these secondary splicing might be post-transcriptional splicing events, yes.
Any more questions?
Maybe I have one. So do you see that the splicing of introns per gene is correlated between individual introns, or does each
there is an enrichment of the same classes within a gene. So, but it's not it's somewhat enrichment, but not very strong. So it seems that it's a bit independent between introns, actually, of the same gene.
And you saw distribution from earlier introns to later introns?
No, not really. So we checked this, but we did not see any bodies.